Good afternoon, ladies and gentlemen. Yes, I just say a few words about my little bit different career path. So yes, I was working in the industry for quite a while, and that's why I defended a little bit late. Let's put it this way. But I have to say that the industry was very much related to the field that I'm researching now. So that was content-wise beneficial. And I think without this kind of a combination, then things would have been a lot harder. Yes, but I will talk about brain research or neuroimaging in particular. And first, a little bit of motivation. So why is it important or worthwhile to study the brain? I think the first thing that usually comes to people's mind is that that's the home for our cognitive abilities, consciousness, and these kind of processes. So it has evoked general interest in people for a really long time. There are, of course, also a lot of philosophical questions that have arisen from brain function, also from brain structure. So what do different parts of the brain do? And the views have, of course, changed over centuries and millennia. Then if we move on to a little bit more practical side, brain disorders are a major expense to the societies. So it has been estimated that in Europe alone, the total cost of brain disorders annually is about 800 billion euros. This is, of course, a lot of money. It's even perhaps a lot more than you can come to think of if you just consider the fact that if we could, through research, just reduce this cost by 1%, so that would release 8 billion euros annually for something else. And this number is about 2 orders of magnitude higher than the investment that we are now currently doing in the neuroscientific research. So I think there's a lot of improvements to be done there, because the gains for the investments in this kind of research they could be so huge. And this is, of course, all not mentioning the human suffering that is associated with these kind of problems. Another point is that the brain is arguably the most complex organ in the human body. This is, of course, as such, not a reason, the complexity as such is not a reason to study the brain. But what I'm trying to say with this is that we say, unfortunately, won't be done by any time soon. So then, how do you actually study, or how can you study the brain? What are the different scales? In space, you can go from synapses, so the connections between two neurons over which the information is transmitted. And then, of course, we can even study the whole body, the brain, plus the peripheral nervous system, muscles, and so forth. And in between, we have the cell level, single neurons. We can study circuits, so how neurons make connections with each other. Then we can study brain regions. There's usually a certain function associated with one region, but not necessarily always like that. And then, of course, we can study the whole brain. So this is the range of spatial scales that we could address. Then we can also look at the timing of brain function. There we can start from microseconds on the transport of certain substances in the brain at the molecular level. Then we can look at the electrical activity. There we start from milliseconds or so. We can move on to seconds and minutes in a typical experiment that we do. But then, of course, we can also study how the brain changes over the lifespan, and of course then talk about years or even decades. What I'm using, and not only using but developing and applying, is non-invasive neuroimaging, as Ari mentioned. So there we usually study structures that are from a few millimeters to the whole brain. And the time scales we can address start from milliseconds and go to some tens of minutes if we think about the span of an experiment. Of course, we can repeat the experiments even in the same person over a period of years and follow the development of the brain and the how certain abilities, for example, language processing develops in the growing brain. So then what are the methods to do this kind of research? I introduced just two. There are a lot more, but these are perhaps the ones that are most widely used and also well represented in Aalto University. We have a variant of this magnetic resonance imaging approach, which is commonly used. Basically, every hospital has this kind of a system. There are about 30,000 of MRI systems in the world. We can use the same technique to also study the function of the brain. So this is called functional MRI. And this is an indirect measure of brain activity. We actually look at blood flow, so how much oxygenated blood is entering certain brain regions. And because the oxygen consumption depends on the activity, so therefore we can indirectly infer something about the brain activity. Now, because of this metabolic indirect connection, this method is slow, so it cannot track brain activity down to milliseconds, as I showed on the previous slide, but we are more talking about a scale of few seconds. It does have high spatial resolution. We can resolve locations on a millimeter scale. Then another method, which Aalto and its predecessor, TKK or HUT, has pioneered, and this is also from which the company emerged where I was working. So that's called magnetoencephalography, or MEG, and that's a direct measure of electric activity in the brain. It is fast, so we can go down to milliseconds. It doesn't have as good spatial resolution as fMRI does. Now, how does MEG work? What are we actually measuring? So if you think that there is a small population of neurons that are electrically active, so there are currents flowing in those neurons, that current will generate currents flowing in the entire head, which then eventually gives rise to a potential difference on the scalp. Now, by sticking just 2 electrodes on the scalp, we can measure the potential difference. So this is very easy. That's called EEG, which is done in every hospital, even in every healthcare center. But there is also a magnetic field associated with these currents. That's also, of course, extremely weak, but with sensitive enough sensors, we can pick it up outside of the head. And now, why do we want to do that? Because it's easy to record the electric potential differences, so why to go for the magnetic field? The nice thing about the magnetic field recording is that we can localize much better the areas that are active in the brain. And this is a typical result from an MEG study. So here the left wrist was stimulated with the tiny electric pulses directly on the nerve, and there is activity in the first somatosensory cortex here. There are 2 different representations of the activity in space, rendered on top of anatomical information of that particular subject. So we get the spatial information. We also get temporal information. So this is the time course of these active spots. So we can see that 22 milliseconds after the application of the stimulus, we have the first response on the cortex. And then there are further responses. And then, of course, we can study how these behave in different contexts when giving different tasks and so forth. Now, MEG is applied not only in research. There is also a clinical application or applications. The main one is diagnostics of epilepsy. So now those epilepsy patients who do not respond well to medication, so that's the first treatment, of course. People are giving anti-epileptic drugs. But about 30% are not, the epilepsy is not controlled by drugs, and then these people might be candidates for surgery. So the epileptogenic cortex will be resected from their brains, and this, of course, requires that we know exactly where these so-called epileptic foci are in the brain. And MEG is a very good tool for that. We can do the measurement non-invasively. We don't need to go into the brain yet. We can do the localization. You can have pictures like this. So there's the anatomy, and then on top of that, this red circle shows the location of epileptogenic tissue. And now this is helpful because prior to the surgery, then the surgeon and the patient can together plan or evaluate whether the operation is worthwhile performing, because sometimes these areas might be so close to so-called eloquent areas that if they are resected, so then there is a severe impairment that results because of the surgery. Also, for those patients who have a brain tumor and that tumor will be removed, it's important to map out those regions that are close to the tumor and which implement some very necessary functions. Then there's a lot of clinical research too. So this is, say, routine use, and then there is also a lot of clinical research. This is certainly not an exhaustive list. It's just a couple of those things that we are also working on here at Aalto University. So predictive diagnostics of Alzheimer's disease. Of course, there is no cure yet for Alzheimer's, but still it would be important to know as early as possible whether the person is going to develop Alzheimer's. Stroke rehabilitation, that's an important thing too, because there's also a large cost associated to cerebral strokes. Then also brain trauma seems to leave marks that we can detect with MEG. And then, of course, there is this neuroscientific research. And towards the end, I will show one example of how you could use MEG for basic neuroscientific research. But before that, so let's look at the actual MEG measurement. Can we actually improve that in some way? And up to now, the only sensor that is sensitive enough to pick up this tiny magnetic field is a so-called superconducting quantum interference device. It's a relatively exotic sensor that requires superconductivity to operate. And because of that requirement, the sensors are housed in a cryogenic environment, which needs to be thermally isolated from the head of the subject or patient. And this thermal insulation, well, it comes with two unfortunate things. It has certain thickness, 2 or 3 centimeters, and then it means that the sensor array has to be fixed. So we can't adapt the array to the head's size and shape of the person to be measured. And if we quantify the distance from the brain to the nearest sensor, so even with an adult brain, this is something like 4.5 centimeters. So we are looking very far from the brain with these sensors. And now if we are measuring a child, so then this distance is of course even larger. Now if we could have these sensors right on the scalp, so if we stay non-invasive, then of course that's the closest we can go, the distance would go to less than half of what it's now with current devices. And now, just a couple of years back, there were discoveries that made it possible to do magnetic sensors that are sensitive enough, which do not require cryogenics at all. And this is an image of one of those sensors. They are rather compact, so it's just 1 by 2 centimeters. And their sensitivity is not quite there of the squids, but they are already good enough. This is a noise spectrum, but I don't go into the details there. So what does it then actually buy if we put the sensors right on the scalp? So this is a simulation study that we performed here in my group. And what you see here plotted on the cortical surface is the ratio, so the gain which you have if you use these on-scalp sensors with respect to the traditional squid-based system. So you can see that many places it's close to 4. In some areas it's even higher. And again, if you had a child to compare with, so then these numbers would be higher. So does it work? That's another question. Okay, just memorize this area is the primary auditory cortex. So we have a gain somewhere between 3 and 4 in that region. So just like 2 or 3 weeks ago, we performed the first measurements here. There's the happy subject, and we had now 2 of those sensors. Here nothing very fancy. We just tape them on the cap like this, and then presented just a short tone pips. And here in blue you can see the brain response measured by the standard commercial squid-based system. Now then, if we do the same measurement in the same person with these novel sensors that are on the scalp, we get the red responses. So if you look at this one, which is above the right auditory cortex, so it's at least a factor of 2 larger response, as was also predicted by the simulations. So okay, so far so good. I mean, we get larger signals, so this is no surprise. But the more important fact is that the spatial resolution improves when we are looking closer. So when we have the sensors closer, we can resolve smaller things in brain activity in space. And this is again a simulation, but now what I'm showing here is the improvement in spatial resolution. And in particular, this is the width of the so-called point spread functions, which are a typical way of quantifying the quality in an imaging setup. So we get factors of, say, from 4 to 6 in this case. And this is a little bit conservative estimate because of the large adult head heads that we have used in the simulation. So now if we have that number and then we compare this to the typical invasive measurement setup that is applied in patients in a hospital, so the epileptic patients, even if they have had MEG, then the surgeons still usually want to implant these corticographic grids onto the brain surface. And they have electrodes which are in a lattice with 10 millimeter spacing between the electrodes. And the information that you can get with this kind of a grid, this is of course important for the epilepsy focus localization, but this is also used for neuroscientific research with the permission of the patient, because they usually have this grid for example, 2 weeks, and then it's boring to lie in the hospital and do nothing, and the recording is just going on all the time, hunting epileptic spikes. But during that time, the patients are usually willing to engage in all kinds of cognitive neuroscientific experiments. So we know that with this kind of a system, we can get information which we cannot access with any non-invasive means. But now if we consider that with the squid-based systems, we can have a spatial resolution of maybe 20 or 30 millimeters. And now if we combine that with the improvement we get with the on-scalp measurements with the new sensors, so factor of 5 or so, we are down to the same kind of resolution as this grid is able to do. So in summary, what I'm trying to say here, that if we have this kind of a system with optimized spatial resolution, we could do something similar what one now needs to do with this kind of a grid in an invasive setup, which of course is only done in patients. So I believe that we will have access to new kind of information about brain activity by this approach. I will now switch gears and describe this neuroscientific approach, which is also novel, unique also in a way that it's the only setup in the world we have here at Aalto, developed by Riitta Häri and myself and many smart colleagues. And this is about imaging brain activity in interaction, in social interaction. And this schematic illustration of interacting persons, I tried to use that to explain why it is necessary to actually study 2 brains if we want to address the brain functions that underlie social interaction. So our behavior, even as a single person, is most of the time proactive. So we are not just passively waiting for the response of the environment and then act accordingly. But instead, we are trying to predict what's going to happen if we perform an action, and then we may adjust our prediction based on the actual outcome of our action. And now if we are in interaction with another person, these so-called action perception loops are fused. So our actions will not only influence our environment and our sensory input, but they will also influence those of the other person that we are interacting with. And likewise, what the other person does, that will have an impact on what we see in our surroundings. So these predictions, in order to work well, they need to involve not only our own actions, but also the actions of the other person. And now, because of this, it makes sense to study the brain processes in these two brains at the same time, rather than just studying one person who is interacting with another person. This studying just one person who is in interaction would be the classical approach in doing neuroscience. So you have the interaction, you measure one brain, you somehow try to annotate or quantify the interaction. Of course, you can videotape it, you can do motion capture, whatever. But you need to interpret that data in order to make sense out of the brain data that you have measured. If you measure two, so then you can combine the brain signals instead of looking at the activity itself. We have a setup with two fMRIs in Aalto. We also have a setup with two MEGs, combining the Aalto MEG and then the Meilahti Hospital MEG system. Looks like this from the subject's point of view. And this is a sample result. We had subjects doing joint movements, and we compute the coherence between the two sets of brain signals. We can see that the motor cortices and visual cortices are coherently active. Just to summarize, so advancing our understanding of brain function, I think it's of general scientific interest. It may yield improvements in clinical practice, cost savings as well. MEG allows us to monitor electric brain activity non-invasively, and this on-scalp approach may bring about game-changing improvements in the signals that we get in MEG in general. And we have this unique hyperscanning setup measuring two persons at the same time to study social interaction. So thank you for the attention.